Hello students, today I am here to discuss about third semester BSc Practical Examination March-April 2023 Practical Scheme CBCS 2021 batch onwards. Okay, subject Biotechnology Practical Paper 3, that is Biomolecules, code BCU.25 301. Okay, here time 4 hours and this carries 25 marks. Question number 1. Estimate the amount of protein in the sample A by Lowry's method or biorate method. Okay. In question number 1, you may get in the examination to perform the practical Lowry's method or maybe a biorate method. Okay. Here, this question carries 7 marks. Here I need to write principle, tabular column itself is a procedure, plot the graph and write the result of unknown concentration. Here in this experiment, Lowry's and biorate method, you are going to get the unknown sample from the examiner, examine the unknown sample and plot the graph. Okay. So standard will be given. Okay, standard will be given here. Okay. Standard OD will be given. You need to plot the graph. So you need to plot the graph, x-axis, y-axis. Put the standard straight line, and what are the unknown sample OD you will get in dotted line to connect so that you will get the unknown concentration. That unknown concentration is nothing but the 7th test tube unknown concentration. You need to write how much microgram you got, that you need to write. Okay. Then next, Lowry's or biorate method are, you may get estimate the amount of reducing sugar present in the sample A by DNS method. Okay, this was a question number 1. So in question number 1, you may get Lowry's method or biorate method or DNS method. In these three, any one methodology you are going to get in the examination. Then question number 2, determine the activity of amylase in the given sample B. Write the principal procedure in detail. And this carries 8 marks. Okay. Here we need to determine the activity of amylase. Okay. So in the given sample, write in detail principle and procedure. Then R, you may get extraction estimation of protein from soaked or sprouted green gram by salting out method. Here also you need to perform, write the principal procedure in detail. Next, question number 3, write the principle and procedure involved in any one of the following. And this carries 4 marks. Okay. So you may get estimation of amino acids by Ninhydrin method. As this carries 4 marks, if you get in the examination Ninhydrin method, write what is this Ninhydrin method and write the Ninhydrin method reaction. Okay, what is the purpose of using this Ninhydrin method? You need to write. You may get separation of plant pigments by paper chromatography. Okay, in these two, any one you may get in the examination. So be prepared for that for 4 marks. For 4 marks, how much you need to write? That much you need to focus on these two, Ninhydrin method or maybe a plant pigment separation by paper chromatography. Then 4th question, describe principle involved in any two of the following. In these, any two you are going to get. That may be TLC, thin layer chromatography, or page polyacrylamide gel electrophoresis, or preparation of buffers, saponification, added number of lipids. For example, TLC here what is this TLC? What is the full form of TLC? What is the purpose of doing this TLC? Methodology you need to write. Okay, and applications. Page, write the full form of the page and where we are using this page and what is this type of electrophoresis, that and applications you write. Then preparation of buffers. First you need to define what is buffer, what are the different types of buffer, and you need to write the applications of the buffer. Then saponification. What is saponification and saponification reaction, applications of saponification. Here the last one, added number of lipids, to write about the added number definition, okay, and the reaction and the applications. Okay. If you are unable to write the reactions, it's okay, no issues. Minimum 5 points you write. Okay. Here you need to write principle involved in any two. Here in these, whatever given the different category of the content. Any two you are going to write. Try to write minimum 5 points, and this carries 3 marks. It means 1.5 marks. For 1.5 marks, 5 points is more than enough. Then practical records, this carries 3 marks. Okay, I hope you understood about the practical examination scheme, about question number 1, 2, 3, 4 and 5. Okay, I'll repeat. Question number 1, you may get Lowry's method or Biorate method or DNS method. Okay. Then question number 2, activity of amylase or maybe a sorting out method by using sprouted green gram. Then question number 3, ninhydrin method or maybe a plant pigment separation by paper chromatography. Then fourth one, TLC or page or preparation of the buffers, then saponification, then iodine number of lipids. Okay. So you need to focus on all these things. Here for the Lowry's and Biorate method, DNS method, this carries 7 marks. Sometimes examiners will give standard OD. If they give standard OD, you need to plot x-axis and y-axis and then plot the standard. Okay, and you need to perform only 2 test tube that thing. If the examiner are not able to give any kind of standard OD, they need to perform all the 6 test tube. Okay. No, you need to perform all the 7 test tubes in that experiments. For example, they may give for the amylase and for this DNS, they may give the same OD. By using that particular OD, you can plot the unknown concentration. Okay. I hope you understood this practical scheme of biomolecules, code DCBTP 301 for biotechnology paper 3. Thank you everyone.